He worked as postdoctoral fellow at National Cancer Center Singapore for 3 years. During his time in NCC, he had successfully reported the predictive factors for genetic screening of BRCA1 and BRCA2. He also reported the importance of genetic testing for 25 breast cancer predisposition genes using NGS technology. Most of his work were reported in PLOS ONE and MPJ Genomic Medicine. This webinar will take around 45 minutes. If you have any questions, feel free to type in the questions field that you see on your screen, and we'll answer them after the presentation. If we didn't get to answer your question during the Q&A session, we'll be sure to take down and we'll email back to you. We'll record down this webinar and will be available on demand on our website after today. Lastly, here's a list of series of past webinars that we have done since April. We will send out the webinar series information after the webinar along with the recording. We will soon have another series of on-demand webinar. Please follow our GeneScript Asia Pacific LinkedIn account and stay tuned for future new resources. Without further ado, let's welcome Dr. Edward to start his presentation. Dr. Edward, over to you.
Hey, thank you, Andrea, for the kind introduction. Good day, everyone. It's a great pleasure to be here on this webinar that organized by GeneScript. I remember a couple of weeks ago, I gave a webinar on how precision mutant library can help in the protein or antibody engineering. And today, I am going to give another introduction about another synthetic tool that offered by GeneScript in order to help in the metabolic pathway engineering. Let me guide you through to the contents of my presentation today. First, I will give a brief introduction about the relationship between the synthetic biology and metabolic engineering, followed by an introduction about our synthetic tool known as the Combinatorial DNA assembly platform. And lastly, I will give a couple of our in-house case study to show you how GeneScript helped our clients in their work using our platform. A little bit of the background. If you think about the biology around you and if you look around at nature, you will see a lot of the beauty and a lot of the diversity. Taking the rose in this picture for an example, you will find a lot of the wonderful details and structure. If you look closely to the petal of this flower, you will find that when you look closely, you will see a lot of things that you have never seen before. You will find that the petal is actually composed of many of the individual cells. And within those cells, there are many little structures that are all giving you a phenotype that you see it as a petal. When you look at the structure like this, everything we see is really about the DNA. Everything that gives us of what we see from the nature, that give us the color, the structure, the function, the things that you think are beautiful and useful, really comes back down to the DNA, which is the hallmark of the synthetic biology. The synthetic biology can be defined in many ways. When I look for the information, I came across this article that published in the Chemical and Engineering News in the year of 2000, in which the synthetic biology was defined as the use of the synthetic capability of organic and biological chemistry to design the non-natural synthetic molecules that has the function in biological systems. That means things that are not from the biological origins and being able to use those chemical molecules to give us the function. As the time goes on, the definition of the synthetic biology has broadened, which is shown here. The design and construction of new biological parts, devices, and systems. The parts, devices, and systems are really those words that come from the engineering. So one of the way we started to think of the use of synthetic biology to redesign of the existing natural biological systems for useful purposes, that is to give a specific application. In this slide, I would like to discuss about how we could apply the engineering principles to the biological systems. By design, for an example, I can build a machine for a specific function, and I know how to draw the way to get there. The modeling is simply about the calculation. That means that we can come up with an equation in order to support the design. The characterization is actually the practice of going through the design, and from what you design to the point where you build at and you test it. And through the testing process, you can characterize the system as a whole as well as the individual part. The abstractions really means actually being able to take now a larger view. And if you go back to the definition of the parts and devices, you need not to go through a specific level of the details, but knowing that I want a bigger function and I can't encode in a simple way. This cartoon shows the engineering in the biology begins with defining the sequence in the natural host, and you can reconstruct of those to give you the synthetic DNA. Now you can have this piece of synthetic DNA to give you a specific function that result in converting the input into a wide variety of the output. Or you can string these devices together, that one has one function and another one has a different function, and compile all of these devices to give you a feature or structures that you are interested in. I would like to switch gear a little bit to talk about the metabolic engineering, which is an area that has been around for quite a while, and we are increasingly seeing the merging of the principle of metabolic engineering and synthetic biology. The metabolic engineering is simply about the biology can do very well in chemistry. That is, from the biological system, you can get a wide variety of the chemical molecules that you want, such as the penicillin, which is an antibiotic, the aspirin, which is a non-steroid anti-inflammatory drug. These two molecules are naturally occurring in the biological system, and they are useful and can give a therapeutic function. Traditionally, when we talk about the synthetic chemistry, it is not that the synthetic chemistry cannot synthesize these two molecules, but the yield is very low, and it's going to take a lot of a step to obtain the final products. Whereas you can have a biological system that can make them rapidly and produce commercially in a large quantity through fermentation. So what's true about these two molecules is they are produced by the organisms that originally make them, and the metabolic engineering is trying to figure a way to improve the natural producer to become better, faster, and more efficient. And the efficiency is simply considered about the yield, that is how much of the input that we provide ends up the products that we want. Here I show you a couple of the examples of the products that produced through the metabolic engineering, including the beer and wine, the bread, the yogurt, the antibiotics, hormones, and anti-cancer drugs. And now the researchers are trying to genetically engineer the microorganism to produce the fuels and chemicals, including the bioethanol, citric acid, and 1,3-propanediol, and so on. So how can the metabolic engineering to improve the natural producers? I look for the information through the social media, the magazine, the article, and I came across this picture or an analogy that best describe about the metabolic engineering in terms of improving the natural producer. What you see here is a maze, and the mouse is trying to figure a way out to get to the destination for the cheese. There are a number of routes that the mouse can use, but we know that not all the routes are productive. So in the field of metabolic engineering, what you need to do is to eliminate those non-productive routes. And what that means is by eliminating those non-productive routes in order to avoid the accumulation of the second metabolites or the formation of the byproducts. Alternatively, you may overexpress the enzymes of the limiting reaction steps, or to improve the enzymes through the protein engineering or CRISPR in order to stimulate or motivate the production rate. And now let's get back to the picture of a cat chasing a mouse. The cat plays a role like a modified enzymes to chase the mouse so that the mouse can run faster to get to the destination for the chase. The third approach is to use the bioengineering approach to turn the microbes into chemical factories. Here I am going to discuss one of the classic examples. In 2018, the researchers have managed to find a way to synthesize the noscapine. The noscapine is a non-narcotic cough suppressant with potential anti-cancer properties. The noscapine is naturally occurring in opium poppy, but we know that it's going to take the opium poppy a longer time to mature before the extraction. Additionally, the opium industry require a control and the restrictive regulation, while the opium poppy can only be legally grown in some area. Another drawback to extract the noscapine is the involvement of a complicated separation processes from numerous of the chemical molecules and narcotic. In 2015, the researchers managed to find a way to synthesize this plant-based drug without having cultivating the real plants. In order to do this, the researchers utilized the genes from the opium poppy, the other plants, and even the rat in order to genetically modify the yeast with more than 30 enzymes. Additionally, the researchers modified the genes and the mediums in which the yeast proliferate, resulting in a massive productions at 18,000-fold. This classic example demonstrated that through the bioengineering approach, it allowed us to reconstitute the natural pathways in heterologous hosts, or the hosts that do not naturally contain the pathway and produce the products economically. Alternatively, the metabolic engineering enables the development of a novel pathway to produce the products in a more direct fashion and more efficient to bring us from point A to point B. And after the development, we can introduce this novel pathway into the cell factories, such as the yeast or the E. coli, in order to massively produce the products that we want. However, the metabolic network in the cell is far more complicated as we thought. Each of the dots here in this metabolic network represent a particular chemical molecule, and the line between the dots represent an enzyme to convert the chemical molecule into something else. What that means is, if I provide a substrate as an input, other than the product that being produced, we will come across the formation of the byproducts. And now we can no longer to knock out the pathways, but we have to think of a different approach in order to deal with the formation of the byproducts, such as to design a control point which allow us to regulate or control how much of the byproducts being produced. When talking about the modulations of the proteins, it is really about how much of the DNA being expressed. That is the use of a different strength of the promoters. Well, now we are actually talking about the modulations at a DNA level. So based on what I described earlier, you can imagine that it is probably easy for the researcher to come out with a proof of principle strength, but it is going to be very challenging for the researchers to come out with a final strength that can meet the yield, the title, and the productivity. Additionally, these final strengths need to attend all biological processes. So it is going to be very costly to progress from the proof of principle strand into the final strand, and it is going to take a longer time. What the researchers are currently looking at is to establish a new technology so that they can reduce or shorten the developmental time in order to promote their products to the market as soon as possible. And here is where our platform, which is known as the Combinatorial DNA Assembly Library platform, can be used in order to help our researchers to speed up their investigation work. Before I continue, I would like to take a minute or two to describe about what is DNA assembly. The DNA assembly is very different from the DNA synthesis. The DNA synthesis is simply putting together all the mononucleotides to form a long oligonucleotide. The DNA synthesis traditionally can be made through the commercial companies. The DNA assembly, on the other hand, are taking those synthesized or cloned genetic part and combining them together in a different organization, changing the order of composition, the orientation of genetic part to build a functional genetic construct. What do we mean by the genetic part? When we talk about the genetic part, it really means about the DNA, the sequences of DNA that can encode the biological function or behavior. There are a number of the genetic parts that we are familiar with, such as the coding DNA sequence that contain the information storage, the promoter that are part of the control transcription initiation, the ribosome binding sites that are the DNA sequences that will be transcribed into the RNA that helps to recruit the RNA ribosomes to initiate the translation. Now, based on the biological data and the engineering principles, these genetic parts can be combined to make a complex unit or designed, and as well as to build a novel biological pathways for different applications, such as to make small molecules to give a therapeutic function, or in food industry, well, it can be used in order to increase the food production. However, we all know that it is always not straightforward to build a new biological system. For an example, if we build a system as shown in this slide, we will never know if it will function properly due to several factors. First, as of now, the genetic circuits that we build are based on our theoretical knowledge. We do not have sufficient practical experience of how it will work on the systems in reality. And second, if you can still remember the complicated metabolic network that I showed previously, we will never know how this newly built gene circuit might interact with the complicated network system in the cell, in which thousands of the components will be involved. And third, even the genetic parts that we use in this gene circuit are not well characterized, such as the strength of the promoters may be different in a different biological systems. In the field of metabolic engineering, a newly constructed metabolic route is imbalanced naturally. And what that means is, if we introduce the newly built metabolic route into the expression system, we may come across several obstacles, such as the accumulation of the intermediates, the generation of the byproducts, the metabolic burden, and even inhibit the growth. Therefore, in order to understand and interpret each of these functions, we need to go through several rounds of the design, build, and test cycles. First, we need to build a prototype of the system and strategically optimize the systems, followed by building multiple versions of the designs, which we normally call it a library of the variants of the systems. And lastly, we test all of these versions to identify the variants that give the best performance. Sometimes we can acquire some knowledge after several rounds of the optimization to achieve a sophisticated performance. There are two different types of the optimization approaches which were published in 2017 in this article. In brief, in the sequential pathway optimization, which was shown on the left panel, a pathway element is varied individually, as indicated as a blue curve, until a local optimum is identified, which is then fixed as a starting point for the optimization of a second element, which was indicated as a red curve, and also the same as the other elements. The combinatorial pathway optimization, on the other hand, as shown in the right panel, it allows us to change multiple elements simultaneously and also enable us to systematically screen the multidimensional space for an optimum. And this very part can comprise of the regulatory elements that include the promoters or the terminator, the coding DNA sequences, the host, the mRNA levels, and different genes homologous. Another difference between the sequential and combinatorial pathway optimization is the combinatorial optimization required very efficient DNA assembly platform. As I show here, the sequential optimization, we can only test less than 10 constructs at a time and can only change one part at a time. In other words, this approach is time-consuming and costly. For combinatorial optimization, we can test hundreds to thousands of the construct in parallel, and we are able to change multiple parts simultaneously. So as you can see that to do that, it allows us to create a large libraries for testing. So, but to do this, by using the traditional gene synthesis and cloning are not sufficient. As I mentioned, because we need to build a large library for variants for the testing. So a more efficient and cost-effective assembly platforms is required. Now, I would like to discuss the three different assembly platforms that have been used among the researchers currently. The Golden Gate assembly is a method that uses the type 2S enzyme to cut and ligate the fragments that we want. This study have shown that it is efficient to assemble more than 5 fragments. However, it has the sequence limitations in which we need to avoid the cutting sites within the fragments. The in vitro homologous free combinations approach is also a commonly used method, but unlike the Golden Gate assembly platform, it has no sequence limitations. The studies have shown that the assembly efficiency drops significantly when trying to assemble more than 5 fragments. Additionally, it is relatively expensive and low throughput. So in order to overcome this limitation, GenScript established our in-house assembly platform that allow us to perform hundreds to thousands of the assemblies simultaneously, and it has no sequence limitation. And because our approach does not utilize the type 2S enzyme to cut and ligate, so the end product has no scar. And what important is our assembly platform allow us to assemble more than 10 fragments at a time. And now I would like to move on to show you a couple of our in-house case study of how our in-house technology can help our clients in their study. The objective of the first case study was to construct a 4-gene metabolic pathway in the yeast to convert a substrate to the precursor of a small molecular drug. The idea was simple. So what the client did initially was to combine all these 4 pathway genes together under the control of 4 constitutive promoters. And then after the construct, the client introduced this genetic circuit into a low copy plasmid origin of the yeast. But the client was not satisfied with the title of the resulting yeast strain because the results showed that it was far from sufficient to reach the commercial production level. So the client was to look for a solution in order to optimize the enzymes for each of the reactions in order to optimize the pathway and improve the efficiency. To do that, the client identified 50 variants for each of the pathway genes through blasting against the database and literature search. And these gene variants were then randomly inserted into each of the coding DNA sequences positioned in the pathway that you can imagine it involves a large library size with different combinations. So by utilizing our in-house technology, we help this client to construct the combinatorial plasmid library. And this library was then transformed into the E. coli and grown on a plate. And to check for the library diversity, the colonies were randomly picked and sequence verified in order to ensure the maximum coverage of the construct being built. All of these plasmid with the inserts were mini prep with the high throughput platform and returned back the end products to the client. So to check the performance of each of their construct, the client was then transformed the plasmid into the yeast cells for performance testing and screening. Here I show you the verification results. To check for the library diversity, as I mentioned, 48 clones were randomly selected for sequence in order to ensure the maximum coverage. From these two tables, you can see that none of the construct was repeated, ensuring little DNA assembly bias. And this slide shows the performance testing data that we received back from our clients. To identify the performance of each of the construct, our client transformed the plasmid into the producing host to perform the high throughput fermentation and using the HPLC to analyze the performance. And they acquired some variants that show a significant improvements in efficiency. And this table demonstrated that the top 24 variants with best performance. The green colored indicated the enzyme variants that were highly enriched among these good performing clones, especially the enzyme B and enzyme C. Taking the enzyme C as an example, the variant 11 and variant 5 appeared very frequently within these 24 clones. In contrast, no particular variant was enriched in the enzyme D, indicating that the enzyme D might not be critical for the performance in this pathway. And now the objective of the second case study was to produce a functional protein in response to a particular environmental signal. We know that the different dose will give a different response, and the expression intensity of a certain promoter may affect the performance of the circuit. Therefore, the initial versions of the gene circuit is very likely to be non-functional. For example, the expression level of the regulator genes may be too strong, and the circuit is always active even without the induction. Or the regulator binding might be too strong to the promoter, making the expression level of the proteins very low. Either way, it is impossible for us to predict the best solution for the design. So in this study, in order to fine-tune the expression level of these three genes, what our client did was she planned to use the promoters with different strengths, ranging from very weak promoter to very strong promoter. Additionally, our client identified two variants for each of these three pathway genes that theoretically comes out with a combination of a library size at 1,000. So again, by utilizing our in-house technology, GenScript helped our client to build each of these 1,000 constructs and assembled individually. And also each of the construct was sequence verified in order to ensure the accuracy and the maximum coverage of the construct that being built, and so that the client can test their clones directly within the library. So here are the performing testing results that we received back from our client. What the client did was to conduct its own test to identify the mutants with the improved traits. So the panels on the left-hand side demonstrated the outcome of the response curve after the induction. And the results showed that the optimized circuit exhibited the stronger and more prompt response behavior than the initial. The results on the right panel show the induced dose response of the promoter 3 strength. And the results show that, as shown in the green color line, the medium strength of the promoter 3 demonstrated the best dose response behavior. Before I finish up my presentation today, I would like to put up this slide to highlight to you the combinatorial DNA library service that offered by GenScript. Currently, GenScript is offering an array combinatorial DNA library platform that allowed our client to construct each of the construct in the library in any of the combination. And using our in-house technology, it enables us to deliver to more than 10,000 sequence verified individual constructs that are ready for the transformation. So thank you very much for your attention, and I welcome for any of the questions.
Thank you, Dr. Edward. So let's move on to the Q&A session. The first question is, what do you think of using the yeast strain for DNA assembly?
Yes, it is very efficient to use the yeast strain in an assembly because it allows you to construct more than 10 fragments at a time and can hold a large DNA molecules. But the drawbacks of using the yeast strain for DNA assembly is it is complicated to manipulate the yeast cells, and many of the laboratories do not have these facilities. Additionally, the growth of the yeast cells is far more slower than using the E. coli.
Thank you very much. Let's move on to the second question. If my library is very large, can I still use the array assembly platform?
Yes, the answer is definitely yes. As mentioned in my slide, that we can help to build up to 10,000 constructs at a time, and we can definitely build more than that. Nevertheless, on the research side, they will need a high throughput screening or testing approach to test the performance, which may not be cost effective. So when the researchers have come across the library sites, come across with a large library site, I would recommend to use a design of experiment workflow to reduce the library size and to concise down the number of the construct, which may seem to be useful for assembly and testing.
Thank you very much. The next question is how synthetic biology is different from genetic engineering?
How synthetic biology? Well, as I mentioned in my previous slide, now currently there are mergings of the principle of the metabolic engineering and the synthetic biology, so that to utilize the advantage of metabolic engineering in order to give us to be used in different applications, such as what I mentioned, to give therapeutic efficiency and the therapeutic function to produce a small molecules, and also can be used in the food industry to produce to increase the food production.
Thank you very much. So the next question is, we know the direction of metabolic engineering, but how to go about it? What are the various tools available?
Okay. As of now, what GenScript is offering is this combinatorial DNA assembly platform in order to help our clients to build a large library size for testing, because we know that in order to improve the metabolic reactions, we need to test each of the functions. So that will come in a great combinations of the use of different genetic parts. So this is one of the synthetic tools that our clients can utilize in order to build a large library size. Another synthetic tool that what GenScript offer is a precision mutant library, as I mentioned in my in my previous webinar. So this precision mutant library enable the clients to introduce the mutations in any part of the regions of interest, so that the clients can test what this mutation does to whether or not it will increase the specificity, the sensitivity, or any of the functions of the library construct.
Thank you very much. Let's move on to the next question. Please elaborate on existing technology from proof to final on slide number 15.
Okay, so what I need to mention, highlight to you that the proof of principles, GenScript do not help our client to design the library construct. So as I mentioned through my, using my our in-house case study, you can find out that the design part is actually done by our client, because we do not know what sort of design that the client want. What GenScript offered to our client is utilizing our platform in order to help our client to build a large library size, so that our clients can test all of these constructs to select the variants that give the best performance.
Thank you very much. The next question is, can I get training for metabolic engineering from you?
Well, this is something that I will probably need to check with our R&D teams, whether or not we can accept an intern on this metabolic engineering part. Yeah.
Thank you. So the next question is how to test in slide number 19.
Test number 19. I believe it number 19. This one. Well, as I mentioned, the testing part is actually well, we currently GenScript do not have the platform to help our client to test the libraries that we built. So clients will have to perform the testing part in order to test each of these constructs that are being built. Yes, this is my answer.
Okay, thank you. And maybe let's move on to the last question. I will note down all the questions we haven't been able to answer during the Q&A session, and send you guys a separate email with the answer from experts. Actually, our speaker, Dr. Edward Wang. So the last question is, are there any planned expression vectors in GenScript list?
Yes, currently we have 3 different expression systems. The E. coli, the yeast, the insect, as well as the mammalian expression systems that are available in GenScript.
Thank you very much, Dr. Edward. Due to the time constraint, we'll end the Q&A session here. For the questions we didn't get to answer just now, I will email the answers from Dr. Edward back to you guys. Along with the webinar recording, we'll send out the APEC webinar series for 2020 that has wrapped up with today's last webinar in another email to you guys. So make sure to follow our GenScript Asia Pacific account, LinkedIn account, to stay tuned for future biomedical news and the resources, and also some of the competition and sponsorship we do for students and researchers. Thank you, Dr. Edward, for the presentation today, and also to all of you who joined us today. I hope you find this session useful and beneficial. Thank you very much for joining us again, and have a nice day.
Thank you very much.
Thank you.